Genetic engineering. Genetic engineering is the transfer of a gene from the cells of one organism to the cells of another. The target can be from the same or a different species. The transferred gene must be able to express itself, meaning it is able to produce its protein. The organism with a gene inserted into its genome is known as a transgenic organism. To give an example, we have this small banana, which has a disease-resistant gene. So if we transfer that gene into a big banana, the resultant offspring can be both big and resistant to disease. A real-life example is the production of human insulin by a transgenic bacterium. First, we need to have the insulin gene from human chromosome 11. Then, we need to cut it out using restriction enzymes. A restriction enzyme is a molecular scissor that can cut DNA into two. So using two restriction enzymes, the insulin gene can be spliced out. If we stretch out the gene, a good representation will be this, with a bit of additional bases on both ends. Do take note that restriction enzymes can cut in a sticky end manner, as shown, or a blunt end manner, where there is no single base sticking out. After cutting out the insulin gene, we need to make more copies of it using polymerase. By adding the insulin gene into a polymerase chain reaction machine, it can produce many copies of the gene, increasing the chance of it being incorporated into a plasmid. Plasmid is bacteria's form of chromosome. Instead of a double helix, bacterial DNA is circular in shape. Using the same restriction enzymes as before, we can cut the plasmid, leaving a gap for DNA ligase to do its job. Ligase is like a glue that can seal the gene into the plasmid. Once the plasmid has the gene inserted, it is known as a recombinant plasmid. And because we produced a lot of copies of the gene of interest, they can also be sealed into other plasmids. After that, we need a host or vector for the plasmid, which is normally a bacterium or virus. To insert the plasmid into a bacterium, heat and electric shock are applied. This process is known as transformation. Once bacteria with successful transformation are identified, it is time to grow them in fermenters. Fermenters are tanks where the growing conditions can be optimized for bacterial growth. The more bacteria with insulin genes there are, the more insulin can be produced and harvested. In summary, restriction enzymes cut out the targeted gene and the plasmid. The gene will be duplicated many times by polymerase. Ligase is then used to seal the gene into the plasmid. Then it is transformed into a vector. What we went through is that the vector is allowed to grow in number inside fermenters before harvesting of the protein produced. If we want the gene to be inserted into the target organism, the vector used is normally a virus, and it will transfect the target organism by inserting the gene inside the target genome. When successful, the target organism will be able to produce the protein, and there are benefits to genetic engineering. Just remember PCC, which is the opposite of CCP. P for the reduction of pollution, C for lower cost, and another C for improved crops. Genetic engineering can produce crops that express toxic proteins, reducing the need for pesticides and preventing harmful bioamplification. Drugs such as insulin can be mass harvested, leading to lower costs. Genetically modified crops can improve food production as they can grow in more areas due to drought-resistant or ability to grow in areas where the soil has more salt. GM crops can also have higher nutritional value, as in the case of golden rice. Despite the benefits, there are ethical concerns. First is the welfare of animals, as animals used in research suffer during experiments. At the individual level, genetic engineering might be how inequality looks like, as it may not be affordable for everyone. There are also concerns about allergies from new proteins produced if the genetically modified animals or plants are meant for consumption. At the commercial level, seeds produced from GM crops might be genetically engineered so that they are unable to germinate. This raises ethical concerns about whether it is right to force poor farmers to buy GM seeds every season. At the national level, genetic engineering research could be a precursor to chemical warfare, and resistance genes might unintentionally pass on to unintended organisms, such as weeds or bacteria, leading to ineffective pesticides or disease treatments.